Hey everyone, and welcome to your Fluid Balance 2 lecture. Let's go ahead and get started. And actually, I'm not even on the right slide, so let me go. Oh no, actually I am. Okay. So for Fluid Balance 2, we are going to list the important sources of acids in the body, describe how protein buffers and the carbonic acid buffer system resist pH changes. We are going to describe the influence of the respiratory system on acid-base balance, describe how the kidneys regulate hydrogen and bicarbonate ion concentration in the blood, distinguish between acidosis and alkalosis resulting from respiratory and metabolic factors, and then describe the importance of respiratory and renal compensations in maintaining acid-base balance. Okay, so the key concept that we're first going to cover is that chemical buffers and respiratory regulation rapidly minimize pH changes. So the four goals of fluid balance and homeostasis is to, one, keep the right amount of fluid in the three fluid compartments, keep the right amount of total dissolved substances in the fluid, so make sure each compartment has the same concentration. Keep the right amount of each individual substance in the fluid, because even though all three fluid compartments have the same total concentration, the concentrations of the individual solutes are going to be different in each compartment. And then we need to keep the right amount of hydrogen ions and bicarbonate ions to maintain normal pH, blood pH. And so this is the part where I would ask questions in class of like, what are the three fluid compartments? They are the intracellular fluid, that's the fluid inside the cell. And then our extracellular fluid, or the fluid outside the cell, is divided into two separate fluid compartments, subcompartments. We have the interstitial fluid, which is the fluid in between cells, and then we have the blood plasma. And then for the right amount of total dissolved substances, what is the osmolality of all three fluid compartments? Each fluid compartment should be about 300 milliosmoles. And then we're going to keep the right amount of each individual substance in the fluid. So look at your table from Fluid Balance 1 and make sure that you know the different concentrations for sodium, calcium, potassium, chloride, and protein. And then the normal blood pH range should be between 7.35 and 7.45. And we'll talk a little bit more about that in this lecture. So proteins function at a specific pH. We've talked many times about the activation of enzymes. So I'm going to go ahead and write a couple things down here, type them out. So it's important that enzymes are a type of protein, that we have activation of enzymes at a certain pH. So one example is that our digestive system, particularly the stomach, the chief cells of the stomach, produce pepsinogen. Pepsinogen does not get activated into its active form pepsin unless it's in a very acidic environment, which is established or achieved by hydrochloric acid. So when pepsinogen is around hydrochloric acid, which is a low pH, it does get activated into the pepsin form, which can then digest proteins. Also, if you remember from B32, if enzymes are outside of their range for normal pH or even normal temperature or other factors, they will start to unravel and fall apart. Remember in Bio 32, you learned about all the different levels of protein folding, primary, secondary, tertiary, quaternary. And if an enzyme is outside of its range for any number of factors, pH being one of them, they are going to start to unravel, and that's called denaturation of enzymes. Those enzymes will denature or start to fall apart. So pH is a scale of hydrogen ion availability and presence. And so make sure you take a look at this scale, and I'm going to go ahead and erase what I just wrote. But the higher the pH, the less hydrogen ions are available. The lower the pH, more acidic, that means more hydrogen ions are available. So I guess make sure you make a note of that. I'll write that over here. Low pH means high, H plus, high pH means low, H plus or hydrogen ion availability. Okay. What is an acid? Ooh, go back to that. What is an acid? We learned about this in Fluid Balance 1, and you learned about it in B32. An acid is a chemical that dissociates in solution, but when it dissociates in solution, a hydrogen ion is one of the things that dissociates or separates from that chemical. So if it gives off a hydrogen ion, it's considered an acid. And we have strong acids, and make sure you know that a strong acid is an acid that completely dissociates in water, so complete dissociation, meaning that it's going to give away all the hydrogens that it has, versus a weak acid, which is only a partial dissociation. So the example that we gave, and I'm going to show you in a picture right now after I spell out dissociation. Here's the example. So hydrochloric acid is a strong acid because it does completely dissociate. It's made up of a hydrogen ion and a chloride ion, and they completely separate from each other in solution. The other example we gave was carbonic acid. Now, carbonic acid has two hydrogens to give, but when it dissociates in solution, it only gives away or releases one of those hydrogen ions, and the other hydrogen ion stays combined or attached on that bicarbonate ion. And so it has two hydrogens to give, but it only releases one of them, so we consider it a weak acid. So let me erase this stuff. We're talking about the next thing. So what is a base? A base is a chemical that unites with hydrogen ions so that it is no longer free floating and able to disrupt reactions. Examples are bicarbonate combines with hydrogen to make carbonic acid. That's one way we get rid of those free floating hydrogen ions, and we use our bicarbonate buffer equation that we'll talk about many times during this lecture. Ammonia combines with hydrogen ions in order to create an ammonium ion. And that's another way to get rid of free-floating hydrogen ions. And then proteins, specifically their side chains, can act as weak bases, and they can combine with hydrogen ions. So make sure you know the examples of different bases that we're going to talk about today. And then pH of blood and body fluid. So we talked about many times before that the normal blood pH range is 7.35 to 7.45. However, at this point, you should know that each fluid compartment actually has its own normal pH or average pH. So the intracellular fluid inside the cells should have a pH of about 7. The interstitial space, and this should already be in your lecture notes, has a pH of about 7.35. And then in the blood plasma, there actually is a difference between arterial and venous blood. If you remember, we've talked about this with respiratory system. If you have a lot of CO2, you're likely to have a lot of hydrogen ions. They go hand in hand because lots of CO2 is going to force the bicarbonate buffer system equation to the right and release more hydrogen ions. So because CO2 is associated with hydrogen ions, venous blood, which has more CO2 content, has a higher partial pressure of CO2, is actually going to be a little bit more acidic because it's going to have a little bit more of those hydrogen ions in its environment. So venous blood is about 7.35. Arterial blood is about 7.4. So still in that normal range, still between 7.35 and 7.45. It's just venous blood has a little bit higher CO2 content and therefore slightly more acidic. So make sure you're familiar with the different pHs of the different fluid compartments. All right, so how are acidic products produced by body metabolism? CO2 leads to carbonic acid by this equation here. CO2 combines with water, which is going to make carbonic acid, which in solution will dissociate or separate into a hydrogen ion and a bicarbonate ion. We have phosphoric and sulfuric acid. That's a result of protein breakdown. Lactic acid from anaerobic respiration. And this is a point in the class where I would ask how many ATP are produced in anaerobic respiration. And you would tell me that it's 2 ATP per glucose. And then fatty acids from breakdown, as well as ketone bodies that are produced when glucose cannot enter the cell in adequate adequate amounts and alternate fuels or fats are burned. So this is something you learn in nutrition, or if we don't have enough glucose available, acetyl-CoA cannot move on to the citric acid cycle or Krebs cycle. And so those get converted into ketone bodies, which are acids, which can make the body acidic if done over the long term. So we have different buffer systems that we use in order to maintain a pH balance. That includes chemical buffers, which are fast acting. They can start working in fractions of a second. Then once those are saturated, we look to our respiratory system to make changes in our breathing rate or respiration rate in order to, again, help balance that pH. But this takes several minutes to start after we've used up all of our chemical buffers. And then lastly, we look to our kidneys, renal mechanisms for long-term pH regulation. And we'll talk about that a little bit later in today's lecture. All right, so I'm going to stop the recording there, and the next video will be on chemical buffers.